I'm a neuroscientist, as some of you know, and I think the aim of this meeting is not to go into any specific details about the projects, but just to give you an idea of how you could use the different facilities. And in particular, one thing I'm going to be emphasizing is why, in the first place, we got stem cell funding for this, and why we think that that's really an opening for making screening. So what the cells that we're interested in, just to remind you of your basic neuroscience, are motor neurons, which here in the spinal cord and will project to all the muscles of your body and make them move. And in several diseases, the one I'm going to talk about, so ALS or Lou Gehrig's disease, what happens is that these degenerate. And this movie actually shows my motor neurons now firing as I walk around the room. Yours slightly less as you sit listening to me. And these are obviously sending action potentials all the way down the axons to the rather grey muscle fibers and making them contract. And in an ALS patient, typically around age 50, but at different stages, what will happen is that these cells will start to atrophy and finally die, actually. So the patients will die within 2 or 3 years. And even before that takes place this process here, where you can see the axons dying back, shrinking back from the muscle. So as soon as that happens, this muscle has no connection with the nerve and it's paralyzed. And what happens in the disease is that paralysis spreads from one muscle around the body until, in typically 2 or 3 years, the patient dies through respiratory insufficiency. And so it's a dreadful disease, and it's just really the opposite end of the spectrum from what Don Landry was saying, is in that we really don't have targets to go after, although I think NF-kappa B could be a very interesting one for the future. And so what we started, especially a few years ago, by thinking is, let's just think about this disease process in simple terms and try and define assays that might help us do something. And so this is that same process in a cartoon. Here is one motor neuron linked to one muscle. There will be many motor neurons for this muscle, and of course multiplied throughout the 400 muscles in the body. The first thing that happens is that axonal dialogue I've shown you, and then, what I didn't show you, there's massive cell death within the spinal cord. So in fact, if you look at the post-mortem spinal cord of an ALS patient, there are basically no motor neurons left, and they're obviously highly paralyzed. So you could think of doing several different things, 2 of which I'm going to show you here. One, obviously, if these motor neurons are not there, you're not going to be able to do anything. It would be good to screen for drugs that prevent motor neuron death, and I'm going to show you 2 types of assays at the beginning and the end of the talk about that. But another way you could think about that is to say that while the nerve cells are still alive and the axon is still there, if we can make this axon regrow and reconnect with the target muscle, then that would certainly have a clinical benefit. And so what we want to do is not so much define drugs, with the exception of the first project I'm going to tell you, but to understand the mechanisms here. This is chemical genetics trying to identify molecular steps which themselves could become therapeutic targets. So wind back the clock about 15 years. The way we reasoned, and Paul Fletcher talked about this in another context, was that if we want drugs to work on motor neurons in patients, we should be screening using motor neurons and not CHO cells. That may be right or wrong in some cases, but so a company I was involved with took these beautiful purified motor neurons and used those as a basis of a drug screen. And so what we did was, from rat embryos to purify enough motor neurons, have survival assays where the motor neurons would die and be kept alive. We successfully identified the lead compound that went into clinical trials, phase 1 and phase 1b. It's been into final clinical trials in ALS. Those have been unsuccessful, like all previous trials, unfortunately. And there's a trial in SMA ongoing. So I'm obviously not here to sell the compound, but this did show us we could go from a basic simple assay in a culture way to clinical trials, but the number of people required to make those primary motor neurons is really just too much to think about in a lab. And so this is where stem cells have really come in. And I think for those of you working on cancer, it's not so apparent that the sheer quantity of cells is a daily struggle for us, because these neurons are post-mitotic, and so making enough of them for a screen can be a major undertaking. And so what Henrik Vischtila and Tom Jessel did here at Columbia was to use the biology of motor neurons, the developmental biology, to develop a way to make motor neurons out of stem cells. And they used a mouse which has green motor neurons because it's got a reporter under the HB9 gene, and using techniques I won't go into at all in detail, but mirroring the developmental biology pathways, they managed to make embryo bodies full of motor neurons. And they could show those were motor neurons in many ways, molecular, but the one neat one was that they could now inject these mouse cells into a chicken embryo in the egg and see that these motor neurons would settle in the spinal cord, and they would actually send out axons along the normal nerves, in fact all the way to the muscles. So these are bona fide motor neurons we can make from embryonic stem cells. The difference is that before, for those primary motor neurons, if you worked for a full day, you might have 100,000 at best at the end of the day. Here you can get 20 million pure cells in an afternoon. And so these have a very high intellectual content for other projects, but simply put, stem cell-derived systems make possible for those of us working on neurons much more high-throughput assays, biochemistry than was ever before possible. So, I'm going to tell you a couple of projects, but this will be the central one. This was done by Heidi before she moved to the screening facility. So this is the second photo you've seen of her today, and she's still hiding in the back, but she'll be the person you interact a lot with for cell-based assays. And in collaboration with Brent Stockwell and Murray Philbin from the beginning, and I'll tell you how we're using the screening facility later. So if you remember, we said one thing we want to do is to make the axons grow back to the targets. So we decided to define a screen for making motor axons longer. Sort of surprisingly, because that's a pretty no-brainer idea, very few such screens have been done. And I think it's mostly because of the absence of the relevant technology. So what Hai did was to take those embryonic stem cell-derived motor neurons that I'll call ESMNs and seed them on a monolayer of CHO cells expressing an inhibitory molecule. It's called MAG for those who are interested, but basically putting them in suboptimal growth conditions, then screening for compounds that could enhance this. And so I'm skimming over what is often the longest phase in a project like this, which is the assay development, giving it enough dynamic range, making it robust enough. And so what Hai did with this complicated two-cell assay with a lot of help from Brent Stockwell's lab was to define an assay in which a benchmarking compound like this would have a very tight dose-response curve. And I just wanted to show the pictures of what I'm going to go back to that. So the way she visualizes the cells is to seed them in 96-well dishes in this case. And since they're fluorescent, they can be live imaged in one of these high-content images. And you can't see too much difference here. But if I zoom into one part of the well, you can see that on CHO cells, regular CHO cells, which are not seen here because they're not fluorescent, the axons in 20 hours grow out quite nicely, whereas on these inhibitory cells, the neurons survive, but they don't put out axons. And so the assay is to try and find compounds that when added to this will make them grow like that. And, so with the benchmarking compound, you can get a nice dose-response curve, and I'm going to go through the screening process, but you can get these nice curves in which what we're here is looking at here is the length of the axons, the blues are the negative controls, so there's not too much scatter there. This is with a positive control, so those are nicely separated. And the green experimental points, this is just a subset of the 50,000 obviously, are mostly inactive, that's good. Some are toxic, that's expected. Some are wildly up here and get the postdoc's blood pressure rising, but they're always just fluorescent compounds. The ones you're interested in are in this compound, in this group here. And so what Hai did, therefore, was to develop this assay, and you've heard this from Chuck, but it's really just to give the message in another way. She did a pilot screen of some 2,000 known compounds, had to go back to the assay to tune some aspects of it, tweak it so it was better. Then we did, together with Brent, a full screen of 50,000 compounds. You're now doing this at 1 well per compound. So you need the assay to be robust. You don't want to throw away too many plates or lose too many active compounds. And then once you have a series of hits, you'll do repeat testing, reorder the compounds, do dose-response curves, and through multiple criteria, including mostly discussing with the chemists, lead selection. And so this brought us down to 4 lead compounds. And so what do you do then? And you do different things when you get those compounds. And so nowadays, we can turn to this army of welcoming and skilled collaborators. So we've heard about the HDS facility. This is the Organic Chemistry Collaborative Center, where Dan and Don and Alison are doing the chemistry you've heard about. And you're about to hear from Brent, I guess, about the chemical probe synthesis facility. So in our case, so we had 4 groups of hits. One were phosphodiesterase inhibitors. You could do a medline and find that out. We didn't do a single thing with that, but that was pretty useful proof of principle, because we know that cyclic AMP is active in this assay. So if we hadn't found them, we might be worried. The second group I'm going to talk to you about briefly, they were known compounds, the statins. And so in that case, what we've been doing is to work with the screening facility to do lower throughput, but still reproducible and repetitive experiments to analyze the mechanism and to find other ways of stimulating this statin pathway. And depending on time, I'll take you through that. Then there were 2 other unknown compounds. That's to say, nothing immediately came up. And just very conveniently, this has fallen into collaborating with the whole range of skills available in the center. So with this particular compound, A, we worked with Dan in particular, who, although we couldn't find the structure initially, looked through the literature and the patents and found a similar compound, which seemed to have a known target. And so what we've been working, which is kinesin spindle protein, and we've been identifying this candidate mechanism, and they've been synthesizing analogs very much in the way that Don described. So there's not time to go into that today, but this really works when you come up with a lead compound. And this second compound here, called B, the structures aren't secret, but they're not informative. We've been working with Rashid and Brent Stockwell. In fact, because it looks very drug-like, it's got a very nice dose-response curve, but there's nothing in the literature about this particular compound. So what they've been doing is to use bioinformatic methods to predict some candidate targets, and that project's not even far enough along to know whether those are the right predictions, but it's a way of getting from the chemistry to some relevant biology, and for us, to potential therapeutic targets. So let me just show you, I'm going to focus mostly on this statin project. I'll probably zoom through it because I want to show you a new type of screen at the end. So when having got through that primary screen, we always say we're never going to get anything from that first set of known drugs, but she did, and it was this well-known cholesterol-lowering drug, simvastatin. And in this case, the chemistry, I didn't have to give Don Landry a penny. We just went to the Sigma catalog and bought the analogs, and you can see that very nicely here, basically all the statins are active, and this is the length of the axon, basically. This is the inhibited cells, and you can see that the very nice dose-response curves going up to a level which I can tell you is completely overcoming inhibition here. And what was striking about these, apart from the fact that this hadn't been reported, was that if we looked at our benchmark compound, the ROCK inhibitor, which you know the one I'm talking about, it certainly works in our assay, but only at concentrations at about 500-fold greater than the statins. So we've been very interested in these, and I'm obviously not going to go through all the biology that Hai has done on this, but just to show you some of the ways we can sort of broaden the assay or tailor it to what we're really looking for. So one of the potential weaknesses of these systems are that they're nevertheless rodent. And if you want to think about going to a human target, you'd like to know whether it works in a human system. And of course, once again, the real difference between the cancer and the neuroscience fields, it was unthinkable. It's impossible to get a human spinal cord neuron in culture to test it. So this is where the embryonic stem cells and the induced iPS cells have come in. This is actually, in collaboration with Kevin Egan at Harvard, we have this fluorescent human ES line that we can make into motor neurons and FACS sort. And so we were able to ask whether these compounds work in a human system. Here's that. They grow very nicely on control CHO cells. If you inhibit them, they respond as well. And if you keep them on these inhibitory cells and add in a statin, they just go wild. They respond even more strongly than the mouse cells. So you get about 6-fold increase in axon growth just in the first 20 hours. So it certainly does work in human models. It works also in the animal. I'm not going to take time to show you the details of that, but it's got many positives. First of all types of nerve cells we've tested so far respond to the statins. And it's nothing special about that MAG inhibitor. Even on laminin, these compounds will have an effect. So this is a general booster for growth. It's good that it's active in this rather artificial model I skipped over of optic nerve crush. We're testing it in ALS mice as we speak. And so these are of potential interest for stimulating either growth of the axons in ALS patients or maybe even following spinal cord injury. But there is a but. In the CNS, if you lower cholesterol, you can't get it from the diet. So if you put in a statin, and this has never really been tested, you may have side effects. So what we've been trying to do is to find ways of stimulating this pathway without having to use the statins themselves. It may seem perverse to have discovered a drug and then to go and look for more compounds, but I think that in the long term, that's going to be really good. In the meanwhile, Brent, and I'm not sure he's going to talk about that, has been doing a lot to improve the availability of these statins in the brain, encountering some problems in terms of half-life. So in this short talk, I'm not going to go into this in detail. What we managed to show was that the statins do act in this system, as they do in the liver, by inhibiting this enzyme, HMG-CoA reductase. So if you want axon growth to be stimulated, you've got to have the risk of reducing cholesterol. So we looked further down the pathway to wonder whether some of these other branches that are cholesterol independent might also provide a way of stimulating the growth. And for reasons I don't have time to go through, we focused on protein prenylation. These, and I think many of you know that prenylation is adding these small lipid moieties to signaling molecules like the Rho GTPases, for instance, to attach them to the membrane to create a local signaling complex. And so what High did was to ask whether, if we block prenylation, do we get the same effect as if by blocking the pathway high up? And just very briefly, she's managed to show that by blocking in parallel all the enzymes that add these lipid moieties to the proteins, she can get the same effects as the statins in the culture. I'm sparing you the primary data here. So what we think we have is a way of, we discovered a new pathway for stimulating this growth, and we now have a way of stimulating that pathway without interfering with cholesterol levels, and so we're trying those things in animals. So that's one way of going from a completely target blind screen, taking a cellular phenotype that's, we think, relevant to the disease, using a cell type that we know is affected in the disease, and getting to compounds and targets that we hope are going to be useful in ALS. But you may have noticed, so stem cell neurons made this possible. We couldn't have done it without, and we've now got 4 distinct mechanisms we're looking into, and I've only told you really about the statins, and these prenylation enzymes, as in cancer, are potentially interesting targets. But it's perhaps striking to you that we've done human and mouse cells, but we actually haven't looked at any ALS neurons, and so what you're going to be trying to do in the end is to treat neurons that have got an ALS genotype or phenotype. And so I'm just going to give you a snapshot in the last 2 minutes of an assay that's been done by Sebastian Tams, who's in the Project ALS lab, and particularly with Hinik Vistula. This is the lab, the human stem cell lab that we both run. And so once again here, the stem cells have made possible something that would be unthinkable using primary neurons. And so what Hinik and Sebastian dreamed up was the idea of actually screening drugs in parallel on control cells, which just express a wild-type version of the disease gene, and this very widely used G93A mutation in superoxide dismutase, which is shown here in green. And these mice develop symptoms that are very close to ALS in lots of ways. And because we could make SOD1 mice, ALS mice that had a GFP reporter, and we could make control mice that had an RFP reporter, and we could derive embryonic stem cells from both of them, then we therefore had these are embryonic bodies, we had a potential source of these neurons, and you can make sure you mix them in equal numbers, and here are therefore control and ALS motor neurons side by side in the screening well, and so if you apply a treatment to them, you can see how each behaves. And so the approach that Hinik and Sebastian took was similar to what Brent's done in some other systems, actually, a sort of synthetic lethal approach, saying first of all, let's use screening not to keep these neurons alive, but to find out molecular details about the specific weaknesses in the ALS cells. And so they screened, here's what you see, the green and the red, and they screened a small collection for the moment, but that's going to increase in size, and looking for toxicity. Obviously, if you find no cells in the well at the end of the culture, that's not highly informative, and so you don't do much with those. But if you can find a stressor that's more toxic for the ALS cells than the controls, then you've maybe started pinpointing mechanisms that could be ALS specific. And just because of time, we can tell you more about this, it's not a secret. I'm just going to show you the results of one of those screens. So you can see here, what we're plotting is the ratio of the wild-type control over the ALS. So if the ALS are more vulnerable, this ratio is going to be higher. And you can see that most compounds have no real significant effect. These are the two controls, either at day 3 or day 4. But here circled are a series of hits that show quite striking different toxicity for the ALS and the controls. And so Hinnick and Sebastian have been following up several of these. They seem to correspond to real mechanisms that are perturbed actually in the ALS neurons. And so I can tell you about that, but I thought I'd just do it for a summary slide. So in where do we go from something like this? Well, first of all, this means that we can actually start screening on ALS motor neurons. Surprisingly, maybe to people from other fields, that's not been done before. But what matters is the results. And now, because we have this approach, we can do the same thing in ALS motor neurons derived from patients, derived from iPS cells that have been made from patient skin. We have a large collection of these. So what I've told you about so far is that the stressors give us some biological insight into the specific defects in motor neurons that have got the ALS genotype. But now we have this assay, which is the ALS motor neurons plus this particular stressor. Then we can argue, I think, that we have a sort of model of ALS in the culture dish. And we can now do a second screen for compounds that will protect the motor neurons against this disease-specific agent, and that's ongoing at the moment. So I think I've thanked everyone as I went through, so thank you for your attention, and I'm on time.